Hi, now let's discuss about cell culture technology. Cell culture is the growth of our cells, animal cells, and or human cells outside of the body and of an intact organism in a cell culture to produce biologically materials such as many more cells or cell-related products such as proteins, DNA, or viruses. Our biotechnological industry is based on cell culture. So this is an important and of practical aspect I want to discuss with you today. So we'll cover cell culture and equipment and reagents and so-called cell lines and primary cells. So what is cell culture? Is the process to grow and maintain cells under very carefully controlled environment outside of the living body. So cell culture is usually related to in vitro experiment. So in vitro makes experiments possible that would otherwise be difficult or impossible in intact organisms. For example, multiple assays in parallel with the same number of cells is very hard to perform in vivo or intact animals. And so by using this in vitro technique, there's a little bit of advantages such as we can perform experiments faster and less expensive than using actual animals. And we can perform much well controlled to draw conclusion. So goal of in vitro experiment is to generate hypotheses and conjecture about what occurs in vivo, which is the most important in the end. So what is hypothesis? Hypothesis is a worldview that is testable. So you can generate hypothesis, which is easier with in vitro experiment, and that clue can be bring into in vivo and hopefully maybe a human or clinical practice. So this in vitro experiment require careful experimental design and also proper interpretation. So we have to keep in mind that during this in vitro experiment, we have to ask ourselves that does a cell in a culture flask dish truly behave like it would do inside in the living body? And many times in vitro experiments, we use a cell line, which is an immortalized cell types, which is you can grow almost indefinitely in the Petri dish. So are these good models for cells that are present in a real body? This is very important question. So let me give you an example. So a cancer cell line, such as HeLa cell, is originated from I guess it's ovarian cancer or cervical cancer from decades ago from one single patient. And that cell becomes transformed into growth in a plastic Petri dish and it's immortalized. So we perform many experiments, for example, testing a new drug, anti-cancer drug, with this cell line. However, we have to know that this cell line only comes from one single patient, while out there are millions of many patients whose cancer, even if it's the same, let's say cervical cancer, that may behave not exactly the same. So we have to know the limitation of when we are using a cell line. So that immortalized cell line is one way of in vitro experiments, but there are some, in fact, many cell types may not have cell line, which means you have to use a cells coming directly from a living tissue that we call primary cell culture. And in that case, that's also in vitro experiment, but there's a little more different aspect we should consider. So first, when we call cell line, that line means those cells have been already manipulated to proliferate indefinitely. and can thus be cultured for long periods of time. So from one generation to the other generation, so we call it as a passage, and it can go as many passages as possible. While primary cell culture coming directly from a living animal usually cannot proliferate indefinitely. You remember the telomere. So cells, when they divide and divide, there's only a finite number of potential to divide. So that's kind of the limitation of primary cell culture. So in fact, there's not so many cell lines. So there I put some examples of commonly used immortalized cell lines. For example, fibroblast. There's a famous 3T3 cell line. Human epithelial cell, it's a cancer cell named as a HeLa cell. Or monkey kidney cos cell, or human embryonic kidney, HEK cell. Or dog kidney epithelial cell, DOCK cell, or CHO cell, which is coming from Chinese hamster ovary. So there are like, many, but still finite number of cell lines established, and we use them for different purposes, such as when we want to study breast cancer, then there's a dozens of breast cancer cell line, which we want to use. If we want to study, let's say, cellular migration from a wound healing purpose, let's say we can use 3T3 fibroblast cell line. So this way we have well characterized those cell lines, and we can compare from one research from another researcher's results. So that serves as a good standard. So let me have a table which compares immortalized cell lines and primary cell cultures. So the advantage of immortalized cell line is it's easier to culture because those cell lines are already transformed, which means it already adapted itself into a well-controlled plastic petri dish environment. So they grow quickly, and also they are relatively homogeneous, and they're fairly well characterized, so standardized, so that we can even compare with others. And also, because of this property, we can grow this cell in a quite big amount. That means we can also manipulate their genes easily. So we can create stable cell lines expressing gene of interest. So by using transgenes or overexpressing or even knock down some of the genes of our interest or not in. In comparison, primary cell culture is supposedly more relevant cell type because it's freshly taken out of the body. So it's more relevant in terms of physiology. And however, the homogeneous aspect, because primary tissue is composed of many different cell types, sometimes it's not as homogeneous when we are using primary cell culture. However, given the primary cell culture, the primary cells are from original living tissue, we can have more confidence extrapolating those results to intact living systems, such as going towards in vivo interpretation. So what are the disadvantages of primary cell culture? Because you took out from the tissue, it's not well characterized, the lifespan is not indefinite, so it has a limited lifespan, such as if you take out cells from the tissue, you may not have a long time to perform your experiment. And also heterogeneous population with high variability. So it may be more difficult to prove something with the primary cell culture. While immortalized cell lines may not have the relevant attributes or functions of normal cells because it's already quite strange cell. And another thing is, even if, let's say, the same HeLa cells, if you are growing the HeLa cell and keep growing and passaging down, depending on that environment, maybe 10 years ago, the published one in the literature using HeLa cell experiment may not be possible, may not be easy to reproduce. Maybe the same HeLa cell, what I'm having, may have changed their characteristics because of over too many passages. The cells may have adapted or changed its characteristics. So that's possible. So one question we can ask ourselves is, hey, we have learned about stem cells. Are stem cells closed immortalized cell lines or primary cell culture? Which is an interesting question to ask ourselves. First of all, stem cells usually are coming from an animal, so in vivo. So it's more like primary cell. On the other hand, stem cell itself has a indefinite self-renewal capability that actually kind of mimics of immortalized cell lines. So one of the proof that the cells we've got this are stem cell is whether they can like keep grow, proliferate to bring a tumor formation called teratoma. So that is one evidence, hey, this is a real stem cell. So in that aspect, it actually also mimics immortalized cell lines. So how to isolate cells from organisms and tissues? There are a number of ways I just want to explain with this picture. So our tissue may come from adult animal or embryo or even egg. So we put them into our petri dish and using blade to dissect them out. But still, you know, our ways of dissection might be too coarse that we could use either enzyme to digest. So using enzyme to dissect them, and then we can have a cell culture. Or very fine chopping makes still, it's kind of small explant of tissue, and that we can actually culture like a primary explant. Or further dissection, if we need, then we can have a small tissue and we call it as even organ culture. So there are various methods of cell isolation depending on whether we are having a tissue chunk or even disintegrate them into an isolated cells by use of enzymes. So when we are culturing cells, the goal is you grow the cells so that we can do experiment with it, or with that we can actually extract RNA, DNA, or proteins. And for that we have to use cell culture and we have to grow cells. So that we call propagation by subculturing. So that's, you can see here. So first we put the cells over here, and then over time it grows. And once it reaches to a certain limit, then we actually subculture, meaning that we took out the cell and we split from one dish into a number, for example, like 3. So that subculture, and we run this on again so that we can exponentially grow the number of cells. So here is a interesting chart with x-axis is number of passages from one generation to the next generation, and y-axis is a log of cumulative cell number. Because you think about it, cells divide 1 to 2, 2 become 4. So it actually undergoes an exponential growth. So when you plot it in a log plot, it will make a kind of a linear graph. So, however, you look at this interesting single passage 1, 2, 3, 4. In the middle is a kind of a dotted line because it's assuming that it can be affected by environment. So, for example, influence of medium. Are there enough nutrients keep coming? Or influence of surface. The surface property. For example, the cells have a contact inhibition property. So normal cells, typically our cells like to attach to some surface. So that's we call anchorage dependence. And if the cells grow too much to spread out to touch itself, they develop contact inhibition. So those can affect this exponential growth. But during this growth, at some point, when the crisis can happen, which means the cells, even originated from primary, can be transformed into an immortalized cell. So that we call as a transformation. And those are the cells we are using as a cell line. So they typically have a characteristic of faster growth and less contact inhibition, and they're likely to generate tumor, so tumorigenicity, so that these cells can be used for a tumor model in animal or in vivo. And they are immortal, which means they don't die over passage. But typical cells, such as primary cells, and mostly they will sometime they enter into senescence, which means they are already programmed to die apoptosis. So now I want to go a little detail of cell culture equipment and reagents. So to supply the cells with the oxygen, nutrients, and growth factors to keep the cells alive, we need to have a growth media. And also we need an environment or equipment to prevent contamination, because ourselves, like animals, we have an active immune system that there are always bacteria getting into our mouth or our body, and we still are okay because we are keep having defense system. But the cell itself has no such such a system. So we have to be very careful that those cells are not contaminated. So for especially the cells are in the culture media, which contains growth media supply, which has nutrients, amino acids, and vitamins, and energy source as a glucose. So it's also good for if there's even bacteria gets in, they will love to grow here. And also we add some serum, which is a blood plasma with clotting factors are removed. So such as BCS, bovine serum. So that is often added, such as 10% into the growth media to culture media. Usually they contain a lot of growth factors, so that can promote survival. However, one thing we have to remember is those serums, in fact, contains undefined mixtures of growth factors and hormones and proteins. So they are good, but sometimes we have to be careful the use of serum when we have to identify a subtle growth comparison because of this unknown effect. So typical cell culture room looks like this, and you can see this is called a biosafety hood and or laminar hood, which there's a filter here, and then the wind is coming from inside to outside, so that it blocks bacteria to getting in. And this is called cell incubator, which keeps the cells, when you incubate the cell, so it keeps humidity and oxygen and CO2 concentration. And you can see some of the cell culture flasks, and there it's not there, but refrigerator, because 4 degree is very low temperature, which keeps down the metabolism. So when we are manipulating the cells, and the water bath to warm up the cells, and microscope to be able to see the cells. So let's go over one by one. This biosafety hood or laminar flow cabinet is to prevent contamination by microorganisms. So it's always blowing inside to outside. And also there's a UV light. So when we are not using it, always turn on UV light so that it sterilizes the exposed surface. And also before and after, we are gonna use 70% ethanol so that, clean up the surface for preventing any contamination too. And cell incubator looks like this, and it's a small housing, and it looks like there's a plate, and underneath there's water which warms up and keep the humidity. So house and store culture flasks or plates, and you have to maintain appropriate temperature, typically 36.5 degree, and humidity and gas concentration to mimic endogenous conditions, so that keep 37 degree and CO2 concentration at a 5%. Also, cell culture, we are using cell culture flask. There are different kinds of flask. One example with a size depending on our needs. So these are called so-called T flask, and depending on the area of the bottom, we can have 12.5 until like 1800. And also, so this is good for like growing the cells, especially like tumors. We can use even like 225 cubic square centimeter, so that we can grow like 10 million cells easily. While for precise assay or precise comparison of treatment versus control, we may want to use multi-weld cell plates. So you can see here 6 weld plate or 12 weld plate, 24 weld plate, or even up to 96 and 384 welds, which gives us a parallel cell assay, which makes sure, because inherent variation is so big that we can have a multiple experiments simultaneously, so that we can get more statistically meaningful and reproducible results. Often many cells need to adhere to a substrate growth, so it depends on cell from cell to cell. So sometimes we have to coat the dish or glass cover slip with an amino acid such as lysine, or extracellular component, collagen or laminin, for specific cell growth. And many times the reagents, we are keeping it in the refrigerator or fridge, which is 4-degree incubator, to keep the cell culture media and other reagents when not used, like we are keeping our food in the refrigerator. And the growth media and antibiotic in culture media, of course, they contain proteins and they can degrade over time, but they can last for weeks if stored in the refrigerator. Also, water bath, often set at 37 degrees, it's to quickly warm cell culture media and other reagents when they directly come in contact with the cells. However, in many times, cancer cells are so strong that many people found we don't even need to warm up the growth media. Still, the cancer cells can survive very well. But it depends on your experiment. So let's say if you are interested in, and the cells are so precious, such as a primary cell, the numbers are so small that you don't want to lose them because of this cold water, then cold culture media, then you have to warm them up. So it prevents certain type of cells being shocked or die by extreme cold temperature of media at 4 degrees, because the cells are optimal at our physiological temperature, 37 degrees. And once you are doing this, you have to watch the cell, whether how the cells grow. So for routine, like every day or every other day, for microscope provides a routine observation of cell culture flasks or plates to inspect the health and confluence of cell. Confluence or confluency means how much covers the cells on the plate. So the percentage of the surface of the bottom of the plate covered by cell. So it's from 0 to 100%. And you can see this, this particular example of T-flask. You watch this and you take pictures of this cell growth every like 6 hours or days. You can see at 72 hours or 3 days after, the cells form and attach them. So this, when they start to attach and grow, they may slow down due to contact inhibition. But that's giving you an idea that, hey, it's time to split the cell from 1 to like 3 or multiple dishes so that you can keep growing. So this ends the cellular principle, and we've talked about building blocks of our human body, cells and tissues, and components of eukaryotic cells, and including cytoskeleton, extracellular matrix, and membrane proteins. And the cell cycle and proliferation and differentiation, especially stem cells, were focused because it has a big importance in biomedical engineering. At the same time, naturally occurring cell death and also as apoptosis. And we also covered about necrosis, which is from the injury or infection or exogenous ways of premature cell death. And the core of this biotechnology depends a lot on the development of cell culture techniques, which allows us to grow cells and tissues outside of the body so that we can get the material in vitro experiments and also a biologically relevant study with enough amount of cells. So thank you for your attention.